Hi, I'm Allie Weber. I'm a postdoc at the University of Washington, and I'm excited to be telling you about some of the work I've been doing in the labs of Tom Daniel and Bing Brunton, investigating the nature of sensory feedback from the wings during insect flight. And I want to start off by showcasing some of these flight behaviors with these beautiful high-speed videos of moths taken by Adrian Smith. Establishing and maintaining stable flight from takeoff, as in these videos, while flying in the wind, while feeding from flowers, is an incredibly challenging task that requires fast and precise sensory feedback. And some of the sensory feedback comes from the wings. And hopefully you can appreciate from these videos that the wings undergo all kinds of complex bending and deformation during flight. So we're interested in understanding how information about those complex patterns of wing bending is encoded by the nervous system, and ultimately how that information is used to guide behavior. We're currently focused on understanding what kind of information is encoded by the sensory neurons that innervate the wings. So I'll provide a little more background on this system. Insect wings are arrayed with sensory structures called campaniform sensilla. And here I'm showing a schematic of the location of these sensory structures on the wings of the hawk moth, Manduca sexta, which is the insect that I study. But these can be found on the wings of any flying insect. And just a quick plug for a talk by my colleague Katie Stanchak, that's part of SICB+, that is about comparative analyses of the distribution and placement of these campaniform sensilla across different species and what the functional consequences of that placement might be. So if we look more closely at these sensory structures, you'll see that these are small domes that are embedded in the cuticle. And each one of these little domes in the cuticle is innervated by a single sensory neuron. So wing bending triggers the opening of mechanosensitive ion channels, which triggers action potentials in the connected sensory neurons. And these sensory structures respond to local deformations of the wing. And so we want to understand what kind of information these sensory structures convey and why they might be located in the positions that we find them. Importantly, previous work has shown that sensory feedback from the wings actually does impact behavior. So we believe this is an important piece of information that the animal is getting in order to stabilize its flight. We are interested in understanding which features of wing movement are encoded by these mechanosensors. So by features of wing movement, I mean, what is the time history of wing bending that triggers a neural response in a particular cell? So just like in the visual system, for example, a neuron might respond specifically to a light decrement followed by a light increment. There's a particular time history of wing bending that might trigger a response in these neurons. And so we're interested in understanding what that stimulus history that triggers a neural response is. So a feature might look something like this, a history of bending over time. And we still have a lot of fundamental questions about how these sensory neurons might encode information. So for example, we don't know whether or not there are different subtypes of these mechanosensors that might respond to different features of the stimulus. We also don't know if these mechanosensors might be found in locations that are particularly informative for conveying feedback during flight. And we also have very little understanding of how information from the wings might be used by downstream circuits to guide behavior. So all of these questions rely on developing a better foundational understanding of what kind of information is encoded by this sensory population. To investigate these questions, we record from the wing nerve using multi-site extracellular electrodes, which allows us to record from multiple neurons at once, while precisely controlling the motion of the wing with a motor that's aligned with the wing hinge. And so then we can use our knowledge of the stimulus that we delivered combined with the responses that we recorded to determine which features of that stimulus are driving neural responses. But there are a few challenges with this approach that previous work has not yet been able to address. So the first is identifying the location on the wing that's innervated by the neurons that you are recording from. So we're recording from the wing nerve, which is a bundle of axons that's innervating a wing, but do not necessarily know which particular location on the wing is connected to the axons that we're recording from. Second, even once you know the location of the sensory neuron that you're recording from on the wing, it's difficult to know what stimulus was experienced at that particular location. So while we can precisely control the movement of the motor at this particular location, if we're recording from a sensor here, for example, we don't necessarily, that stimulus is going to be transformed as it propagates along the wing, and we don't necessarily know what is being experienced by the sensory neuron at this particular location. Okay, so to address this first challenge of identifying the location on the wing that we're recording from, we turn to a method developed by Pratt et al. Earlier, using an infrared laser to do focal heating. And that local heating of a particular campaniform sensillum can alter the firing rate of that cell. Typically, it reduces the firing rate, but it can also increase the firing rate. But so this gives us a way to focally target a single location on the wing to identify whether or not that corresponds to the location that we're recording from. So this slide is showing an example of a case where we're recording from a particular neuron. The location is unknown on the wing, and when we turn the stimulus on, we see an increase in firing rate, followed by a decrease in firing rate when the stimulus turns off, as expected. That's with the IR laser off. If we now turn the IR laser on, and if we're positioned over the location of the sensory structure that is innervated by the neuron we're recording from, now when we turn that IR laser on, we get focal inactivation at that particular location. And so we see a clear reduction in the firing rate when we have the IR laser targeted over that particular campaniform sensillum. And this actually allows us to localize the location that we're recording from within a few hundred microns, well under a millimeter. So now on to the second challenge, determining the stimulus at the particular location on the wing that we're recording from. So we know that these sensory neurons respond to strain on the wing. So in other words, they respond to the local bending at their particular location, not just the displacement of the wing or its movement up and down. Yet, all that we know from the setup, the basic setup that we have, is the motion of the wing at the particular location that we're controlling it. So to address this gap, we can record from different locations on the wing using these laser displacement sensors that allow us to track the motion of the wing at those locations within a few microns of accuracy. And then using multiple sets of those recordings at specific locations, we can reconstruct the full surface of the wing with very high temporal precision. Here in a moment, I'll show you what one of these reconstructions looks like. Keep in mind, this is not going to look like a natural wing flapping motion. This is the reconstruction of the wing movement during the stimulus that we're presenting. And this is something like a white noise stimulus. So we're trying to present a large range of possible frequencies and bending patterns that will allow us to better determine which subset of those are triggering responses in neurons. So here's what this reconstruction looks like. So the whole loop will be 30 milliseconds. So there's a lot of complex bending the wing is undergoing on very fast timescales here. And what this allows us to do is estimate the local deformation and from that the local strain at any particular location on the wing. So now we can use that reconstruction to determine which features of wing bending are driving responses in these sensory neurons. So what I'm showing on the left here is similar to previous work that has been done, where we're simply using the displacement of the motor to estimate what the neuron is responding to. So in this plot, the vertical axis is the stimulus displacement, just at the location of the motor. And with that, we can estimate a time history of that displacement that the neuron is responding to. On the left, we're using that wing reconstruction to estimate the strain at the particular location that that sensory neuron is innervating on the wing. And so now we have a time history of strain at that location rather than displacement at an arbitrary location that that neuron is sensitive to. And in both cases, there are some similar commonalities to these two features. So both show a very short latency of the response within 10 milliseconds, and both show sensitivity to some fluctuation of that stimulus. But overall, these features look quite different depending on whether you estimate that feature with the stimulus displacement at the motor or with the reconstructed strain. And so this shows us that knowing the location that's innervated by that neuron is really incredibly important for determining the feature that the neuron is most sensitive to. So I've shown you some new methods that we're using to determine which features of wing movement are driving neural responses. So I showed you an example of an individual mechanosensor that's selected for a particular stimulus feature. I also just want to briefly mention, I didn't have time to show this work in this talk, but we've also shown that individual mechanosensors are actually selected for multiple features of the stimulus. And so if you're interested in that, then check out the poster listed on this slide. I also showed you how we're using wing reconstruction to reveal the features of strain specifically that neurons are sensitive to. So moving forward, we can use these techniques to answer a variety of questions in the system. So for example, we can assess whether there are distinct populations of mechanosensors that might respond to different types of temporal features. We can also determine if there are particular modes of wing bending that are particularly well encoded by the system, given the location of the sensors on the wing and the types of features that they respond to. And we can also develop a better understanding of the information available to the animal under different stimulus conditions, including under more naturalistic stimulus conditions where the wing is undergoing a more realistic trajectory as it would during flight. And so all of this is moving towards a better understanding of the sensory information that is available to the animal during flight, which will eventually allow us to understand how this information might be processed by downstream circuits and how it might guide the behavior of the animal. With that, I'd like to thank all the members of the Daniel and Brunton labs who have helped out with this, particularly my advisors, Tom Daniel and Bing Brunton. And a big thank you to Abby von Hagel and Lucie Wolf, who are co-authors on this project as well. Abby is a post-baccalaureate researcher at the University of Washington, and Lucie Wolf is a high school summer student who worked with us. And both of them collected some of the electrophysiology data that you saw, and also some of the wing tracking data. So a big thank you to both of them. Thanks for attending my talk. If you have more questions, I'll be available via Pathable, or feel free to reach out to me by email. My contact information is on the slide here. Thanks.